We are here at the EOR2C NCI AACR Symposium on Molecular Targets and Cancer Therapeutics with a panel of experts to discuss the following topic: cancer clinical trials, the need for a new algorithm in the age of stratified medicine.
My name is Stefan Sleijfer. I'm a medical oncologist from the Erasmus MC in Rotterdam, the Netherlands, and I represent the EOR2C as one of the scientific co-chairs of this meeting.
I'm Ken Anderson from Dana-Farber Cancer Institute in Boston. I'm one of the scientific co-chairs of the meeting, and I represent the American Association of Cancer Research.
Hi, I'm Jim Doroshow from the National Cancer Institute, also one of the co-chairs.
Roger Stupp, EOR2C President, medical oncologist from Lausanne University and designated director of the Zurich Cancer Center.
Gentlemen, thank you very much for taking the time to talk to European perspectives. The current model for cancer drug development involves an investment of in excess of $2 million and a timeline that may take up to 12 years from concept to clinic. Is this sustainable? I'll start with you, Professor Anderson.
Tragically, I think it not sustainable. And this meeting is representative of the explosion in science, the unprecedented knowledge in genomics that's really defining the underpinnings of cancer. And so these discoveries that will be reported here need to be exploited, if you will, or translated, as we say, from the bench to the bedside to benefit diagnosis, prognosis, and treatment of our patients. And honestly, the number of new targets and the number of new validated targeted therapies is unprecedented. So the opportunities for and the promise for our patients has never been greater. But in order for this to happen, the model you just mentioned of excessive cost, and you didn't mention it, but it's also 7 to 10 years time, really isn't acceptable. So we will hear at this meeting, and many of us are excited about making newer models where we can more rapidly translate than the time that currently is required, and at a much lower cost. This meeting represents an incredible collaboration, an international collaboration, not only from the three groups represented here, but from investigators around the world that span those that are necessary. Those doing primary discovery to those doing the clinical trial and the science of clinical trials. Industry and biotech are well represented at this meeting, and all of this is inspired by our patients. So this meeting will go a long way to remedying that problem that you mentioned. We can use the science to better inform clinical trials, do the right patients at the right time, targeted therapies, shorter trials, less cost, delivering on the promise of science for our patients much more rapidly.
Professor Sleijfer, what do you think from the European perspective in relation to our current model?
No, I fully agree with Professor Anderson that this current model is not sustainable. Taking the Netherlands as an example, where I come from, so I know the situation there a little, we have seen that over the last years the healthcare costs have increased with almost 6% each year. And currently the healthcare costs approximately cover 12% of the gross national income, and it is expected that it will raise to approximately 20% in 2020. Part of these increasing costs are, of course, because of the aging of the population, but also the introduction of new technologies and new, but also very expensive new drugs. So we really have to find ways to make those drugs more cost-effective to dampen these massive increases in healthcare costs.
Professor Stupp, the failure rate of a lot of drugs that look good in preclinical development probably approaches 90%. How do we improve our conversion of promising candidates into effective drugs?
I think we need to take the human experiment earlier on. I think we need to have clinical trial designs which answer biological questions and not only clinical questions. These need to be integrated. We need to test new molecules early on as proof of principle in very selected and smartly designed clinical trials if we want to know what we take forward. we probably should sometimes a little bit temper our enthusiasm for new agents before we go into large-scale clinical trials, and possibly also through better collaboration avoid some of the redundance when the same target is repeatedly tested with similar compounds by different pharmaceutical companies.
Dr. Doroshow, what are your thoughts in relation to how we really get the right drugs to the right patients?
I think I can just exemplify that issue by talking a little bit about how we've reconfigured our early therapeutics program. It's actually was just yesterday approved by the NCI's Board of Scientific Advisors, a reconfiguration that will focus primarily, number one, on having a network of institutions and not simply individual institutions, no longer doing phase 1 trials where we're looking for endpoints like toxicity, but rather only proof of mechanism trials. We basically decided not to sponsor any early phase trials that don't allow us to obtain tissue and to do proof of mechanism studies. We think that will move us along much faster, allow us to actually test more drugs in a smaller number of patients to understand what is happening, as was said, early on in the process, and then to focus more clearly on those drugs that seem to be most promising from a molecular and a clinical perspective combined.
That's a very exciting development. Professor Anderson, there's been suggestion that many of our trials we only see incremental benefit when we should really be looking for transformational benefit. How do we move from incremental to transformational oncology?
I think that, again, the opportunity to do that has never been greater than now. And the way you do it is by actually understanding better the underpinnings of cancer, in particular the genomic abnormalities that are characteristic of a particular patient's cancer at a particular point in time. And so this meeting is going to be emblematic of that again. We can, as Dr. Doroshow just said, we can define targets and we can examine patient tissues and show that we've actually hit the target with a given novel targeted therapy and see what the clinical impact is. But we can actually do more than that and, if you will, make it transformative rather than incremental, because this meeting and elsewhere we can determine now the opportunities to combine targeted therapies predicated upon very strong scientific evidence for blocking, if you will, multiple pathways concomitantly. And you know, that's the way empirically cancer was cured years ago, childhood leukemia, Hodgkin's, testicular cancer, by empirically combining drugs. But what this meeting most excitingly represents is the opportunity for transformative science, which then transforms to effective combinations of targeted therapies appropriate for the right patient in the right context. So I do think we should expect and we should hold ourselves to the standard of transformative rather than incremental science.
Professor Stupp, you talked earlier on about getting more information earlier in trials and perhaps looking at the use of phase 0 trials or looking for more activity in phase 1. How do we do that?
I think tissue is the issue. And I think getting the tissue, and I think this is often called optional, and I think this should be mandatory. And any trial we do, if we don't verify the target, if we don't verify what we have been doing when we treat our patients, I think is doomed to fail. So we need to try to understand why something works or doesn't work. So yes, here there are opportunities. The challenge is sometimes to find these windows where sometimes for the individual patient there is not a clear, tangible benefit evident. But on the long run, it is going to be beneficial to everybody. If we verify the targets, we go rational. I think what we just heard, also combining targets, and here I would think the regulators are asked, at least in Europe, we are often not allowed to use several experimental drugs at the same time. And here maybe cooperative groups, here regulators, here different models of research can help to bring the different new combinations forward. But again, it needs verification early on whether we actually do what we presume to do.
Professor Sleijfer, we've already touched there on the regulatory aspects of clinical trials, and we do see significant differences between the US and Europe. Health technology assessment will be one example, but there are others. Do we need to bring the regulatory agencies closer together? And if so, what type of model should we follow?
It would be great if we could achieve that, in particular in terms of bringing the expertise together with respect to the efficacy and toxicity parameters. But of course, we also have to be realistic, and health technology assessment also covers cost effectiveness of drugs. And of course, there are very great differences between the countries in terms of reimbursements and what is cost effective. And also within Europe, we have not been able to manage that across Europe, so I don't think it's realistic to get that done for both Europe and the US. But still, I think there's a great opportunity to join the forces with respect to efficacy and toxicity parameters.
And Dr. Doroshow, in relation to the FDA and the EMA, what ways do you think they can work more closely together for mutual benefit?
Well, they actually do work fairly closely together, and I know because I participated that there are regular phone calls, monthly interactions between the appropriate offices at the FDA and the appropriate counterparts in Europe. It seems to me that one of the things that often doesn't happen, however, is that academic institutions in different parts of the world, because of some of the regulatory barriers, find it difficult to cooperate across large geographical boundaries. And as we move into this worldview in which we're looking for smaller and smaller subsets of patients, the U.S. is not big enough. In terms of the patients we need to search for particular subsets of diseases. So we really do need to be able to make it easier to ship drugs back and forth across the Atlantic. That sometimes is a challenge. And so that we can screen patients on a national and international basis to move studies along more quickly.
Professor Anderson, you talked about combining different approaches, and certainly we've seen in myeloma how that's been very advantageous. How do we do that in the context of different drugs being used by different companies or developed by different companies? What sort of partnership do we need to develop to really make that move forward?
You know, that's a wonderful question and a very key issue if truthfully we here and others are going to deliver on the promise for our patients. Because we can make seminal discoveries, we can have validated targets. As Dr. Doroshow said, we can cooperate around the world and recruit the appropriate patients for the right trials. But honestly, having even an FDA-approved medicine or an EMEA-approved medicine does not guarantee access, nor does it guarantee, as your question implies, that we can use the right combinations. And so the people that are necessary to get the process done are the biotech and pharma, the academics, the regulators, the funders of cancer research, and then most importantly, our heroes and inspiration, the patients. But I think I mention that primarily because if we remember that what we're all here for is to help our patients, then the bridges will be built automatically. So let me go right to your question. If you have competing companies that happen to have medicines that we know are better together, synergistic than when used alone, but we as representatives of scientists here on the panel have evidence to suggest that they're useful, it's our job to help bridge that gap and remind everyone that the goal is to help patients. Honestly, if we do that, if we deliver on the promise and get the targeted therapies in combination to patients who need them, everybody will win. It will be a major success for biotech and pharma. So the model of competing with each other is wrong. The model of competing with cancer is correct.
An excellent way of putting it, Professor Anderson. Professor Stupp, most of our concentration in cancer clinical trials and also in terms of cancer budgets tends to be on drugs. Do you think we maybe overemphasize that and we should also be looking at new approaches in relation to surgery, radiotherapy in relation to cancer clinical trials?
So surely you're right to the point. I think we should not forget there are other ways of combating cancer with the other modalities. which for mainly funding reasons is not appropriately investigated. I would say there is not investigation, but it's not to the same extent and not with the same way. And I think along what we just heard, I think here again collaboration is needed. And yes, and if we want to look at least in solid tumors, the progress made over the last 2 decades is when we combined, let's say, radiation and chemotherapy, and now radiation and targeted drugs. And so yes, here there need to be more emphasis. Here we need to have new models of funding, because as there is no one producer who will invest and get the revenue, we need to find other platforms. And I think the academia, cooperative groups are the answer to that. Also along how to collaborate among competing industries and companies. Here, an academic network, as it was mentioned, whether it is national or international, and transatlantic as it becomes more and more, is the model to go forward. And I think the answer would be short: yes, we need to investigate on surgery, also on radiation, on combination thereof. We need to clearly decrease the regulatory barriers for all this collaboration when we want to have it. And it's true, in principle, we all serve the same goal, but sometimes we are limited by the fact on clinical trials everything is much more difficult to do and to be funded than we do it when we do the same thing outside clinical trials. So here I think the academia is ready to go for. Now the regulators, the insurance companies need to follow if we really want to make substantial progress.
Dr. Doroshow, you touched on this a little bit earlier as well in relation to sort of the reconfiguration that NCI has done over the last number of years. Could you maybe share with us briefly just some of the aspects of the new clinical trials network, and also maybe what we can learn in Europe from that?
So the focus of what we've done really is to, as you've heard before, is to promote collaboration. And never before, at least in the U.S., as we funded our large cooperative group enterprises, they were all funded separately. We had 10 different organizations, all competing tooth and nail, often for the same study. The reconfiguration basically will now reward individuals specifically for collaboration in terms of their funding. It will provide more resources for the patient care costs and research costs involved. And most importantly, I think, will provide an underpinning both in the area of IT. So this is, I think, really perhaps of the things that have been done has not been well. People don't know about this, but in fact, as we sit here today, the U.S. now has a single IT clinical trials data management system that has been implemented in 3,000 sites. All of our trials, the cooperative group late phase studies, the early phase trials, our specialty networks, all will be using the same software. Which I think is going to be an important way to improve productivity and to also allow and stimulate the ability for individuals, wherever they are, to participate in trials everywhere. So that no longer, since we will have a national network, all of the trials will be open to every investigator at every site. And I think that's probably the biggest change that never occurred before, but will allow us, I think, to complete studies much more quickly, which is really our goal. Our goal is not to open studies, but it's actually to complete studies.
Absolutely. I think that's a powerful way to move forward, and certainly the IT infrastructure that you mentioned is also very important. And Professor Sleijfer, obviously the area that we've tended to concentrate a lot on as a prime focus of this meeting is in relation to translational medicine. One of the things that we've touched upon earlier in relation to clinical trials is the importance of tissue samples. How do we ensure that biological samples and biological endpoints are embedded into clinical trials?
To make it mandatory, and in particular for early clinical phase studies such as phase 0 and phase 1, it is really necessary that we understand what we are doing and that the drug that we are exploring is indeed doing what we expect it should do. For example, if we are focusing on a drug that is inhibiting a specific target, we really want to see also in those early clinical trials that that drug is indeed inhibiting the function of that protein. So it requires tissue, so it should be mandatory in the context of those kind of studies.
And in relation to candidates, there are probably now 1,000 candidates in various forms of development for cancer clinical trials. How do we best, Professor Anderson, separate the wheat from the chaff? So how do we drop the non-runners early while translating those with promise into effective therapies?
Yeah, so I think that's a super question because there are more options now than ever before. I think we need to have, as much as we can, good preclinical models. We have the genomics at unprecedented levels at the present time, so we can define targets. But we need to use the preclinical model systems to test not only individual targets, but also combinations. And then, honestly, we need to advance only those that look most promising in the context in which they look promising. And I would just mention that in the United States, the Food and Drug Administration has actually been very proactive in this way in a new field called regulatory science, where we're really applying the principles of science to drug regulatory processes. But for example, there is the ability now to go forward with 2 novel agents, neither of which has gone forward individually, but which is suggested and predicated upon preclinical data, suggesting that the combination of novel agents is what should go forward, not the individual agents. And you can do that without doing the long, arduous process of each one individually. But to get right to your question about how do we define those that are effective, that's very important. But equally important in my view is what I call successful failure. That means demonstrating early that something doesn't work. So to be honest, as Professor Sleijfer just said, tissue samples are essential at the time of diagnosis for the purpose of treating the right patients and the right subgroups. Tissue samples are very important in patients on protocols who are responding and those who are not, because we can learn in that latter instance what is the mechanism that is conferring resistance. That can often lead to abandonment of that strategy, or even more importantly, to then build on that. If you hit the target and you still have another pathway that's conferring resistance, that tissue sample can serve as the basis for a subsequent combination trial. So it comes down, I think, to tissue sampling and doing very informed, scientifically based, outcome-based clinical trials.
Dr. Doroshow, some critics are suggesting that certain personalized medicines approaches are overhyped and that actually the benefits are minimal, while the costs are particularly high. What do you say to that?
Well, I think that sometimes the costs are high, and we've just very recently in the U.S. seen one of our major cancer institutes actually decide not to take on a approved agent because it was not deemed to be any more effective than other alternatives. And we'll probably see more and more of that kind of thinking, because I think it's appropriate to think very clearly about the benefits of any of the new agents that we're looking after. I think it's also clear that there really are only a very few targeted agents that have clearly demonstrated a survival benefit. So in that context, that criticism can be viewed as, I think, very appropriate. It doesn't mean that the approach is wrong. What it means is that we need to work harder in finding out how and in what context we should use some of these medications that are so much more well suited to particular molecular contexts. I think the issue really is, a long time ago there was a paper published by our colleague Dr. Dan von Hoff, the title of which was "There are no bad drugs, just bad clinical trials." And if you think about it, we're embarked in actually a process of thinking very hard, especially over the last decade, about what we could learn from the exceptional responses that occurred in targeted therapy studies, which 5%, 2%, 8% of patients responded in a particular disease. The drug was written off for that disease, but sometimes those patients had very, very dramatic remissions. The issue is when those studies were done, we didn't have the tools to understand why those specific patients might have responded. And we're going back to look at this enormous database of retrospective information, but to look at it prospectively in terms of can we determine the molecular characteristics that led to the response in those so-called failed trials. Perhaps there's no such thing as a failed trial. It's just our failed ability at the time to interrogate the information that is now potential.
Professor Sleijfer, in relation to translational medicine, one of the key things we've realized is that companion diagnostics is an integral part of personalized medicine. Already the FDA are linking approval of new innovative drugs and their companion diagnostic. Are we doing the same in Europe?
No, not yet, but I think and I hope it's only a matter of time. It's clear that when you have a drug that is working in a specific population, and a population can be identified by a certain genetic aberration in tumor samples, then it's also obvious that you need a good assay, a reliable assay to determine whether or not that specific aberration is present in the tumor. And if you don't have such a test, then such a drug will not work that well outside the context of clinical trials. So it's clear that you need reliable and robust tests that should meet certain criteria before implementation of such a drug and such a test in the clinical daily practice.
And Professor Stupp, how do we get the companion diagnostics reimbursed? Because there are issues about this in Europe and in different countries. What are your thoughts on this from an ERTC perspective?
I'm not sure there is an ERTC perspective on it, but it's clear some of the challenges, how to understand how we reimburse value something brings, whether it's the drugs, the drug pricing is arbitrary, and the same is for the companion diagnostics. Now, many of the companion diagnostics are developed together with the drugs. They go along. So some of it will be the reimbursement together. Or I could just give it back to Paul and say, how to reimburse any other diagnostic test, and it should be just reimbursed as we do for any other diagnostic test. Is it really too expensive to pay $2,000 or $3,000 for a companion diagnostic if we can save $25,000 or $30,000 or euros of costs of an ineffective drug? And we don't only save cost of the drug, but we also save toxicity, we save time of the patient, false hope, and so on. So this is an issue I think we can deal with. The big challenge is all these companion diagnostics need validation, and it needs prospective validation, and this needs an integrated approach. And we come back, we need tissue, we need the solid database. You can't do that all retrospectively. We have to agree to disagree, to do it one way and not 5,000 different tests who are slightly different. And here I think the challenges are with us to have some discipline and also to go forward with what we have and to stay with the tool rather than changing the tool every single day.
Professor Anderson, the academia-pharma intersect is a very important area. In fact, The Oncologist has a particular section in the journal devoted to it. How do we encourage increased collaboration between academia, biotech, and the pharma sectors, particularly in the areas of cancer drug development and cancer clinical trials?
Yeah, that's a wonderful question, because to be very frank, unless that relationship and collaboration continues and actually grows, the opportunity to deliver on the promise of all of the scientific discoveries really will go wasting. I do think that our model in academia, funded in large part by the generous support of the National Cancer Institute, academia makes a fair amount of discoveries. But it's been very frustrating over the years because the model system is you have a new finding and then by chance maybe a small biotech or a pharma may or may not have a therapy directed at that target. And if it isn't the case, then there is this process that goes on of either a new biotech takes up the charge or a big pharma decides that this might be a profitable target. But what I'm getting at is that it's a team again. You cannot in academia really go all the way to the bedside, at least not in our present system. And honestly, as it's become more and more obvious, big pharma really does need the research expertise that is going on funded by the National Cancer Institute, but at large cancer centers and otherwise in academia. So we need to foster the relationship. I think, again, if we remember that the goal here is to help patients, and that if we understand the process, it's quite a natural relationship. The fear I have, quite honestly, is there's a large, what's called conflict of interest concern, which I am completely sensitive to and very sympathetic with. But what it can't do is separate that relationship. If I had to point to one thing, to be just to get to right to the point, that has been responsible for the advances in cancer that we've seen so remarkably in the last decade in the blood cancers and solid tumors, and the promise that's represented at this meeting, it is the fact that we have that relationship between academia and industry. This meeting exemplifies that. There are scientists and clinicians here from academia and industry, and one of the wonderful aspects of this meeting is surely the formal presentations and the didactic, but much, much more important are the interactions that happen at this meeting, and honestly, the collaborations that result, the new clinical trials that go forward, again, delivering on the promise for patients. So I think we need to do everything possible to foster the relationship where neither academia nor the biotech and pharma alone is enough. We need to be together if we're going to actually get it right for patients.
Okay, final question, and I want you to be brief, less than a minute each, okay? In order to be truly transformational in cancer medicine, what do you think is the key thing we need to do? I'll start with you, Professor Sleijfer.
Well, very briefly, I think we should focus on drugs from which we really understand the mechanism of action. And that allows us at a very early stage in the clinical drug development to see whether or not that drug will be effective.
Dr. Doroshow?
I think we really need to set a very high bar at a molecular and a clinical level and take things forward that we can understand in patients.
Professor Stupp?
I think we need to emphasize collaboration, as it was said all along this roundtable, across academics, across nations, but also between industry and academia. We are partners, and it's not the good and the bad guys, and I think we don't exploit these opportunities for collaboration in a sufficient way.
And finally, Professor Anderson?
We have to have very high standards for transformative targets, and we need to have team science and collaboration.
Gentlemen, thank you for what's been a wonderful round table and giving us some real insight into the area of cancer drug discovery, development, and the identification of molecular targets. Thank you very much for your time.
Thank you.